Let's start with anaphase now. So here ana means up, phase means stage. So during this anaphase, chromosomes are moving up, that is towards two opposite poles of the cell. Hence it is called as anaphase. So let's study what happens inside the cell during this anaphase. So during this anaphase, at first centromere splits. Centromere of the chromosome splits. Centromere splits. So actually by the end of metaphase, what happens? All the chromosomes align at the center of the cell, forming equatorial plate or metaphasic plate. Now in the early anaphase, centromere splits, dividing the chromatids of each chromosomes. So after splitting of centromere, chromatids of chromosome start moving towards two opposite poles of the cell. Chromatids of each chromosome start moving. They start moving towards two opposite poles of the cell. Towards two opposite poles of the cell. Two opposite poles of the cell. So let's see how they are moving, and during this movement how they appear. So this is the animal cell membrane. This is the cell membrane of animal cell. Okay, and then these are the chromatids which are moving towards two opposite poles of the cell. So this is centromere and they are joined at kinetochores. Like this, they are moving towards two opposite poles of the cell. Okay, like this they are moving. So this is centrosome. Inside the centrosome, we know centrioles are present, which are perpendicular to each other. Okay, so during this movement of chromosomes, centromere is towards the leading edge. Centromere is at the leading edge. Here also centromere is at the leading edge, whereas arms are trailing behind the centromere. So during the movement of chromatids, centromere is towards the edge. Centromere is at leading edge, whereas the arms are trailing behind the centromere. Arms are trailed behind the centromere. Okay, now these chromatids are called daughter chromosomes. These chromatids are called as future daughter chromosomes because these daughter chromosomes, these chromatids become the part of daughter nucleus. So these chromatids are the future daughter chromosomes. Okay, now let's see how these chromatids are moving towards two opposite poles of the cell. So the movement of chromatids towards two opposite poles of the cell occurs with the help of both interzonal fibers and chromosomal fibers. Chromosomal spindle fibers and interzonal fibers. So movement of chromatids or chromosomes. So chromosome with one chromatid is also called as one chromosome. So movement of chromatids or chromosomes, towards two opposite poles of the cell occurs because of the expansion of interzonal fibers expansion of interzonal fibers. I'll explain what are these interzonal fibers. Interzonal fibers, and because of the dissolution of chromosomal fibers. Dissolution of chromosomal fibers. Chromosomal fibers. Okay, let's see how they are moving with the help of these two types of fibers. So now these fibers are called as chromosomal fibers. Chromosomal fibers are the spindle fibers which are attached to the chromosomes at the point of kinetochores. So chromosomal fibers and interzonal fibers. Interzonal fibers are the spindle fibers which are formed in between the chromatids after their separation. So after the splitting of centromere, chromatids of a chromosome start separating from one another and they start moving towards two opposite poles of the cell. When the chromatids of a chromosome separates from one another, interzonal fibers are formed between the chromatids of each chromosome. These are called interzonal fibers. Now these interzonal fibers starts expanding. So due to the expansion, they start expanding. Due to the expansion of these interzonal fibers, these chromatids are pushed towards two opposite poles of the cell. And also these chromosomal fibers starts dissoluting. Due to the dissolution of these chromosomal fibers also, these chromatids are pulled towards two opposite poles of the cell. So these interzonal fibers are providing, they are pushing the chromatids towards two opposite poles, whereas chromosomal fibers are pulling the chromatids towards two opposite poles of the cell. So now chromatids are moving towards two opposite poles of the cell by both pushing and pulling. Both pushing and pulling. They are both pushed and pulled towards two opposite poles of the cell. And by the end of anaphase, they reach towards two opposite poles of the cell. And during this movement, chromosomes or the chromatids appear in different shapes depending upon the position of centromere present in these chromosomes. Now let's study the shapes of chromosomes which appear during anaphase during their movement towards two opposite poles of the cell. Okay, so anaphase is the best phase to study the shape of chromosomes, whereas metaphase is the best phase to study the number of chromosomes and morphology of chromosomes. So now let's study the shapes of chromosomes present in the cell during this anaphase, that is during the movement of chromosomes towards two opposite poles of the cell. So anaphase is the best phase to study the shape of chromosomes. Okay, so depending upon the position of centromeres, these chromatids or the chromosomes appear in different shapes during their movement towards two opposite poles of the cell. So here, depending upon the position of centromere, these chromosomes are of four different types. They are metacentric chromosomes, submetacentric chromosomes, acrocentric and telocentric chromosomes. Now, metacentric chromosome. Metacentric chromosome means a chromosome which consists of centromere exactly at its center. That is called metacentric chromosome. So now this is metacentric chromosome. After splitting of centromere, these chromatids like this, they start moving. So centromere is towards the leading edge, whereas arms are trailed behind the centromere like this. Like this they appear. So they appear V-shaped during their movement, during the anaphasic movement of chromosomes in case of metacentric chromosomes. Next, coming to submetacentric chromosome. Submetacentric chromosome is a chromosome which consists of centromere near the end. Near the end. That is just away from the center. Near the end. So this is centromere. So after splitting of the centromere of the submetacentric chromosome, like this they appear. So this is the centromere which is towards the leading edge. which is towards the leading edge. So these chromosomes during their movement they appear L-shaped. L-shaped. Next, coming to acrocentric chromosome. So in case of acrocentric chromosomes, the centromere is present, is very close to the end of the, it is very close to the end of the chromosome. So like this, it is very close to the end of the chromosome. So after splitting of this centromere, now this is at leading edge, centromere is at leading edge, and these are the arms like this, and they appear arms which are trailed behind, and they appear J-shaped. 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 Next, coming to telocentric chromosome. In case of telocentric chromosomes, telocentric, telo means end. The centromere is completely at the end of the chromosome. Here, close to the end. Here, near the end. Here, at the center, exactly at the center of the chromosome, near the end of the chromosome, and very close to the end of the chromosome, here at the end. At the end. In telocentric, centromere is present at the end of the chromosome. So this is the centromere. After splitting of this centromere, now this is the centromere which is present at the leading edge, and these arms, only one arm, and they appear I-shaped during their movement. During their movement towards two opposite poles of the cell. In case of human cells, telocentric chromosomes are absent. We cannot see telocentric chromosomes in case of human cell. So this is all about anaphase. So what happens in the early anaphase? Centromere splits. After splitting of centromere, chromatids of chromosomes start moving towards two opposite poles, and they move towards two opposite poles with the help of both interzonal fibers and the chromosomal fibers. Due to the expansion of interzonal fibers, they are pushed towards two opposite poles of the cell. Due to the dissolution of chromosomal fibers, they are pulled towards two opposite poles of the cell. So here chromosomes are both pushed and pulled towards two opposite poles of the cell during anaphase. And during their movement, they appear in different shapes. So anaphase is the best phase to study the shapes of chromosomes, whereas metaphase is the best phase to study the number and morphology of chromosomes. So depending upon the position of centromere present in chromosomes, these chromosomes appear in different shapes. Metacentric, submetacentric, acrocentric and telocentric. Metacentric chromosome means V-shaped, submetacentric L-shaped, and acrocentric J-shaped, telocentric I-shaped. Like this, by the end of anaphase, by late anaphase, all these chromosomes or chromatids, chromosomes containing single chromatid, reach towards two opposite poles of the cell. So let's say this is the animal cell. So in animal cell, they reach towards two opposite poles of the cell. Okay, so anaphase is the phase where the chromosomal number is doubled, because due to splitting of centromere in anaphase, each chromosome is with only one chromatid. Suppose if the cell enters into cell cycle having 10 chromosomes, the number of chromosomes present during anaphase will be 20. So the number of chromosomes gets doubled during anaphase due to splitting of centromere. So this point we need to remember. This is the important point during anaphase. So this is all about anaphase. Next, let's start with telophase.